So thank you, Margarita and Nick and the organisers for the invitation to speak to you today. It's a real pleasure to be here. So I'm going to be talking about some of the work that we've been doing with those Cambridge rats, looking at compulsive like checking using this novel animal model of the observing response task. Now, I'm going to begin actually by acknowledging the huge amount of people that have contributed to this work. So Trevor, who you've just seen, has been collaborating on this work since the beginning. There's a whole array of people who have contributed to this task within the group. And we are a translational lab, so we have some people in the group working exclusively with humans. You'll hear from Margarita after the break about her work. But we also have a large number of people in the lab working with the rats, including Marina, who is sat in the audience, who's been running this task. And in fact, we'll be back in the lab running it again tomorrow. So it's a real team effort. I'll also thank the funders, including Sir Garden, who have funded some of this work as well. Now, why would we want to use animal models to understand OCD, and how can we use animal models to understand OCD? The key advantage of using animal models is that it allows us to address questions about mechanism, because we have control over the experience of those animals. So it means that we can investigate questions like, for example, the effects of early life stress and the subsequent development of OCD and compulsive-like behaviour, which of course it wouldn't be ethical to do in humans. It means that we can go and look directly in the brains of these animals. After we've characterised their behaviour, we can look for changes in particular brain structures and look for differences in the shape and the size of neurons that we wouldn't be able to do in humans. It allows us to test novel drug treatments, and using some of the more advanced imaging technologies, we can actually look in the brains of these animals as they are performing behaviour. So we can look in the brains of these animals as they are checking. Alongside that, we can also directly manipulate brain regions, so we can turn regions on or off by taking direct interventions into the brain as and when we need to for the purposes of the experiments. There are many reasons why it would be good to use an animal model to test underlying mechanisms in OCD. Now that begs the question of how can you try to model a condition like OCD in animals? And I think the key thing here is that we don't try to model the entire condition. So of course the way that we diagnose OCD in the clinic is that we have essentially a checklist of symptoms that need to be satisfied in order for a diagnosis to be given. And so if someone satisfies that list of symptoms, then they will be diagnosed with OCD, whereas someone else with only some of those symptoms would not be. Now, that's the way that we diagnose OCD clinically, but if we're thinking about the underlying psychological and biological mechanisms, actually this checklist of symptoms may not be really what's happening in the brain. Those sort of, the boxes that we put people into may not reflect what's happening at the level of the brain, because we know that, for example, people can have the same diagnosis and yet express that mental health condition in a number of different ways. There's more than one way to get a diagnosis of OCD. So if we think about the underlying brain processes and psychological processes, we might identify different processes that may actually be relevant to multiple mental health conditions, and that people would exist along a spectrum for each of these psychological processes. So we all have a particular psychological fingerprint of all the different psychological processes that we have. And people will vary along this spectrum. People with the same diagnosis may vary along the spectrum in different ways. But the key thing is that a particular combination of factors would be the fingerprint that would lead ultimately to this diagnosis of OCD. So that then, if we think about the condition in that way, allows us to then focus in on some of these and to try and just simply model those aspects of the condition. So if we focus in on compulsivity, for example, that allows us to develop animal models that focus on just one of these processes and to characterise individuals that show this behaviour and those that don't. So we're not trying to model the whole condition, we're really focusing in on specific parts to make the model more tractable. And the particular behaviour that we focused in on for the observing response task is checking. So as Trevor's just introduced to you, we have our Cambridge rats that we are training to work for rewarding food pellets. So these are sucrose pellets. They're highly motivated to work for these rewards. the animals are trained in such a way that they are presented with a bit of a dilemma. If they press one of two levers, they will gain their food pellet. But the correct response changes unpredictably throughout the task. And the way in which we ask them to respond, it's not immediately obvious which is the correct response, even if they are working on the right lever. So it's quite a challenge, even for these very bright Cambridge rats. So to help them out, we give them access to a third lever on the back of the chamber, which is termed the observing lever. And this will illuminate a light above the lever that is currently being rewarded. So what the majority of our animals will do is use that information to guide their behaviour, and they will then go and work on that lever. And I should just point out that the cue is only on for a very short period of time. It's just there to give the information as to what the currently correct response is. The lever will still continue to be rewarded even once that information has gone, until it switches again. Now, one of the really nice things about this task, besides the fact that it is there's a parallel task that can be conducted in humans, which was developed by Sharon Moraine-Zamir, who is in the audience, so you can grab her at coffee and quiz her on that. Alongside the fact that we have a human version of the task is the fact that we can separate out these two versions of checking behaviour. So we can see the difference between what we deem functional checking, which provides information about the reward and adaptively guides the behaviour. This is guiding the animal how to gain more rewards. And we can separate this from what we term dysfunctional or perseverative checking, where the animals, a subpopulation of animals, will continue to press despite the fact that the information is already there. So there's no further information provided by this behaviour. It's excessive, it's perseverative, and it actually takes time away from where they could be directing their behaviour towards the levers that would actually give them the reward. Now, we have multiple variations of this task that allow us to address very specific questions about checking. So we can make the task even more uncertain and look at the impact that that would have on checking. And what we see there is that, as you would predict, in fact, human studies would back this up as well, the more uncertain the environment, the more the whole population checks. So that's an increase in functional checking across everyone. Interestingly, our subpopulation, who I'm going to talk about shortly, continue to show increases in excessive checking alongside that as well. So this backs up this idea that functional checking is adaptive. Now, we can also challenge the animals by raising the stakes. So if, alongside not getting a reward, they actually have a risk of getting an electric shock for making the wrong response, that's also a further incentive to increase checking. And again, we see an increase in checking, this functional checking behaviour, when we bring in this punishment if they get the response wrong. And in that subpopulation of animals, we also see an increase in dysfunctional checking. What's particularly interesting is that in our particular subpopulation that we think are relevant to OCD, they don't bring their behaviour back down once it's safe again. If we remove the shock, they don't change their behaviour, which is really interesting. We can also change the task such that the cue doesn't give any information anymore. Rather than illuminating above the correct lever, we just make a light in the middle of the two levers illuminate. So there's no information there. And these animals, this subpopulation that we're really interested in, continue to check despite that not providing any more information. And finally, we have a new version of the task that we've developed in the lab where we actually directly punish checking. And some of our OCD relevant population will continue to check even though they will be punished for doing so. So those are the variations of the task. These are the kinds of questions that we can address with an animal model. What have we learned from this task? And I'm going to highlight really 3 separate studies that we've performed, but we have more. One thing that we've been particularly interested in from the psychological level is this relationship between checking and habitual behaviour. So when we think about why the animals are pressing this observing lever, it could be that they're doing this in a very goal-directed way. They want the outcome of knowing which lever will be rewarded, and so they make the response that will give them that outcome. They have this representation that they want that information. But there's another reason why the animals might make that response, and it could be simply that the lever is there. It acts as a stimulus response habit. And so seeing the lever drives them to respond, even though they're not really representing that outcome. Now, one thing that we've been really interested in is characterising how stimuli in the environment play into this kind of habitual behaviour. So one way that we can look at this is to look at the ability of cues in the environment to capture behaviour in a really unconscious way. So an example of this might be, and we will see at the break whether this plays out, that we may have within our environment an array of stimuli. We may have an array of cues that might guide the way that we react. And if we're in a very kind of goal-directed, acting in a goal-directed manner, we may have an outcome that we want to achieve, which is eating healthily, in which case we should make the response of choosing the banana. However, if there are really high-value alternative cues there, like a delicious-looking cupcake, if you are very prone to these stimuli grabbing your attention and directing your behaviour, what you might find is the response is quite different, and instead you go and choose the cupcake instead. Now, we can characterise this in a much more formal way using tasks that essentially determine how easily reward cues grab your attention. And one such task which Sharon's lab have been running is called value-modulated attentional capture, which is just a very fancy way of saying how distractible are you by these cues. Essentially, what you have is a target detection task. You're looking for a bar of a particular orientation, but some of the distracting cues have been paired with rewards in the past. So if you are highly distractible, it will take you longer to find that target because you're paying more attention to the cues that were paired with the reward. And so when Mike Lepelli's group in Australia looked at this and compared it to the BATCAP OCD score, they found there was a correlation, a positive correlation, between the influence that these cues have on behaviour, so how much these cues capture your attention, and OCD symptoms. Now, we can actually look at this in rats as well, and this is the subpopulation that I've been referring to, because we can run an analogous task in rats, so a procedure called auto-shaping, where they don't have to do anything, we just present them with a cue and a reward. And their behaviour makes no difference to the delivery of the cue, but we find that there are individual variations in how animals respond to this task. So some animals, when you present them with the cue, will go to the reward location and wait there for their reward to be delivered. And we term those goal trackers because they're going to the goal location and they're tracking that. But we have a subpopulation of animals who, instead of doing that, go to the location where the cue is presented. They're being really driven by that cue, and that's determining their behaviour, and we deem them sign trackers. And it's that sign tracking population who are the group that show this excessive and perseverative checking. So consistent with an increased control of environmental cues on behaviour, it's these animals that show the high sort of reliance on habits and these high levels of excessive checking. And that's one of the most replicable results I've ever seen in the lab. I think we're now on 15 replications and 10 different experimenters have seen this same effect. So we are convinced that this is real. So we have a psychological set of results that we can get from the observing response task. We can also use it to try and understand the brain regions that support functional and dysfunctional checking. So rat brains are obviously simpler than human brains, but there are analogies between the two. So these are both pictures of the brain from the side, so sort of through the middle of the head. And the colour coding is meant to represent that there are analogous areas to the anterior cingulate area that Trevor was just talking about and to the supplementary motor area. And we know from work originally done in Trevor's lab that we can lesion particular structures within this network, so we can take out the dorsal striatum, analogous to the chordatum putamen, and see decreases in functional and dysfunctional checking. We can target regions of the prefrontal cortex, again, which Trevor mentioned, and that leads to increases in functional checking and reductions in, no changes in dysfunctional checking. And we can target regions like the nucleus accumbens, a structure that feeds into the chordate and the putamen, and there we can see increases in functional and dysfunctional checking. Now, more recent work within the lab is focusing on the role of dopamine in these regions. And some recent work done by Louise Pickenham in my group has shown that if we target the nucleus accumbens and we target the expression of dopamine pharmacologically directly within the accumbens by doing intrabrain injections, increasing dopamine within the nucleus accumbens increases functional checking but leaves dysfunctional unaffected. But decreasing dopamine within the accumbens decreases both functional and dysfunctional checking, and we're just following those up now. So we can use the ORT in these animal models to understand some of the neural mechanisms, the neural basis of this checking behaviour. The other advantage of these animal models is that it provides a platform for testing novel medications. So I'm glad Trevor has introduced MRS because that means I don't have to. But essentially, basing on the large body of literature that Trevor talked about, including the study conducted by Marjan Biria, we know that there are these differences in glutamate and GABA ratios within the anterior cingulate, which, as Trevor explained, motivated this desire to look at reducing metabotropic glutamate signaling, reducing the amount of presynaptic release of glutamate by targeting these mGlu-R receptors. And you've just seen these data, but we do see reductions in dysfunctional checking behaviour with both a positive allosteric modulator of these receptors, so this flexible one that helps these receptors out, and by directly targeting them as well with that Lilly drug. So we can use the ORT as a platform for testing novel medications, and we now have a lot of future plans, actually developed in consultation with people with lived experience, so I'm hoping that these will chime with you. We have a number of plans going forward for the ORT and for this work, and we have a grant application in at the moment where we're hoping to really kind of dig into those brain circuits, really understand the networks that support functional and dysfunctional checking, and whether we can drive functional and dysfunctional checking, both increases and decreases, as a proof of concept by changing activity within those circuits. We also want to know how those drugs targeting mGlu-Rs are working. We have a very strong hypothesis that this is likely to be acting on these regions that Trevor talked about, anterior cingulate or supplementary motor area, but we need evidence for that, and that evidence is probably best gained by direct interventions within animal models. And finally, we would like to take a more kind of treatment-focused approach as well. As Trevor said, we don't know why SSRIs have effects in OCD. We can test that in this model, and we can do that both at the whole animal level, but also by direct brain interventions. And we want to know how SSRIs and their effects on these circuits differs from the mGlu-R treatments. So I will stop there, but thank you very much for your attention.